If you want to start your own company, you can't really just say, I have a technology, I have a scientific finding, and I want to move it to the next level. If that's what you want to do, likely it's best to stay within the confines of an academic laboratory or even work in an industry laboratory. But if you have a project, if you have something larger that you can accomplish that not only is something that's of pharmaceutical value or of financial value, but something that actually brings value to the research community as well, that's when you can really operate as a separate institute. So being a startup in the life sciences industry, I think the lesson that I've gotten from working at so many is it's not good to have a startup that's just one project, because how is that really different or better than a laboratory, right? If you have a whole company that's about one RNAi technology, for example, or one diagnostic platform, this isn't particularly different from actually just continuing to do that research in your laboratory. And I think what we've been able to do is do a surprisingly large amount of work without doing much except focusing on what we can do best and bringing together partners. Because in the end, those projects, any project that's worth it, any project that adds value is going to be larger than the sum of its parts. And so, does anybody have any questions? Yeah.
So, how do you exactly find the molecule that you want?
So it's not clear to me, how do you find the molecule? How do you develop the molecule? Do you work with a lab that has already found some target, and then from there you develop the drug, or how exactly does that work?
So if you look here, in every single one of these boxes, this is basically our process. In every single one of these boxes, somebody has to take care of it, right? And so what we do is, it can be that somebody has a target and we find a molecule, right? It can be that somebody has a molecule and then we advance it. The point is, we don't look at it that way. We look at it that there is a larger task that has to be accomplished, right? And how many different groups can we take, including ourselves, to fill in all of the parts? And once it's filled in, you can actually accomplish that. So let me give you an example. Do you want to explain kind of the example of the ETSU collaboration? Because that's how we got to that molecule.
So basically, in this case, this project ARLS Mine, we met a PI. He found us, actually, and so he had a target in mind, and he studied depression his whole career, and so transitioning to becoming a medicinal chemist would be quite daunting for him. So he reached a point in his project where he realized he could design a new drug, but didn't have any of the skills to do it as he identified this target. So we're working with him to design a new drug to fit his target that he's got in mind. And so, from our perspective, this is a great collaboration, both because he has advanced knowledge of a target and he has very particular assays that are perfect for characterizing antidepressants.
And so here's a second example, and it's completely at odds with the other one, yet they really work in the same exact framework. We work with a professor at Texas A&M University. We work with this professor, we have collaborative grant submissions. So he's on a multi-institutional R01 going out tomorrow. 4 years, almost $2 million. And we've worked with him to get preliminary data and doing some blinded independent analysis of some of our work, right? So we've engaged him in that route. We came up with the molecule. We invented this molecule in our own laboratories, and we brought it to him for independent testing, and now we wrote this grant together. See, in that way, we added value to his research, and we were able to create essentially a whole project out of nothing, right? Nothing but intellectual capital. And so hopefully that answers it a little bit.
So do you get funding from venture capital, or just where do you get your funding from?
So far, our funding is from private investment. And we also hope to get additional funding through grant sources, and we're also in talks with larger venture groups. But generally, venture capital wants to come in at a later stage in the pharmaceutical industry. And our model is more that we can leverage academic research in the basic preclinical end of it. And so we may not even want to look at venture capital because they would want to take a very large slice of it, and creative control is important at this part of it.
work? Like, what do you guys keep versus the institution, and how does that?
So we are an institution. What's nice, I think I have to say, I said a lot of bad stuff about, and a lot of difficulties about, when do you want to start a startup? What are the reasons you don't want to do it? But the really positive aspect of it is that we are an institution. We have every single, this is America. We have 100% of the same rights as MD Anderson, as Texas A&M, as Rice University when it comes to intellectual property. So we keep our IP, all of it, and they'll keep their IP. This is just how it is. And that's the real advantage to starting your own company. Because in our laboratory, our work is, we derive the value from our work. And that's really what I hope everyone in this room wants for themselves. If you're here because you want to learn more about startups, you're probably here because you want to derive more value from your scientific research. And I couldn't agree with you more. And that is the real benefit to starting your own company. It is worth those challenges because I think precisely what you just said. Precisely because we don't have to be concerned about the IP. Of course, we're concerned about the IP. When you bring on institutions, we have agreements and that we'll sign, right? We have NDAs, for example, we'll protect our compounds by blinding them. We'll call them compound A, compound B. And if our partners invent something during the course of it, well, it's theirs. We don't see this pre-negotiation of who invented what as a good approach, because it limits the kind of creativity that you can go through. And everyone is very incentivized to make deals because this is medicine and it's a team sport.
So what keeps you up at night in the current state?
What keeps me up at night? Yeah, what keeps me up at night is literally what's been keeping us at night is writing grants, doing work, not being able to sleep because we have so much to do. But what worries me is, are we going to be able to get our continued funding, right? That's one thing, right? That's always a problem a startup's going to have. But actually, our funding has been accelerating as our results accelerate. So we've been really blessed on that. Recently, we've had a lot of interest in our company because we've just come up with a novel antibiotic candidate with one of our partners. And so I think that what keeps me up at night is trying to make sure I can get to the next threshold. Can I open up another pipeline? Can I get somebody to sign another agreement with me? Can I check off the boxes that my really larger potential investors want me to check off before they'll put in $1 million? These are the types of things that keep me up. I am not worried about IP because in the end, who invented what? That's just going to wash out. That's a reality. I'm just worried about doing the research. We're worried about trying to create value. And the fact is that you can't be slowed down by the potential concerns because that's just never going to work in a startup. So all of my normal anxieties, I have to try to just shed them. And I think it's worked, So I try to just concern myself with getting money and getting to the next threshold.
So if I can, I'd actually take a little bit here. So first, let me introduce myself. So I'm Corbett Miller. I work for the Arrowhead Drug Shop, so I'm the chief scientist. I did my PhD here in Houston at Rice, and I just finished a postdoc at Yale and came back to join Drug Shop. And so to kind of get at your question of what keeps us up at night, it's actually very similar to probably most professors before they have tenure. It's getting data to get funding. And so for most professors, they realize there's a timeline before the university decides essentially to fire them or to give them tenure or to let them stay on and so forth. And for us, it's actually very similar. So we have to get data, and we have to get data to get funding, and at a certain point, our funding's going to run out, and then it becomes like a feedback loop. If you don't have any money, you can't get data, and you're done. Much like being a professor.
But instead of publishing and perishing, just to add to that, what's really great is that kind of what we would maybe call vanity doesn't really play a part, because Dr. Miller, you were able to when you told our investors about our progress and showed them our progress, what was their response? They didn't need to see it published, right?
Yeah, so, it's a different audience. So for us, a lot of for our private investors, they're not in a panel reviewing your data in detail. They need to be convinced that your idea is sound and that it's going to work. You have to explain it's a different audience, a very different set of criteria for what's important. But that said, we do apply for grants as well, so we do both.
So what kind of timeline are we talking about? Is you're recording? Please everyone speak into the microphone.
Please wait until I give you a microphone. Thank you.
So, I don't know if this is on. So what kind of timeline are we talking about if I have a drug target that I want to develop a small molecule for? And what are the fail-safe mechanism that you get into it? Because in academic research, there's a lot of leeway where things fail and there's less, I would say, accountability financially. So for you guys, maybe the threshold is very high there. So what are the fail-safe mechanisms that you have to use to sort of overcome those kind of problems?
Sure. So to answer your first question, what timeline would it be to partner with us, I guess, if you had a target? We have, to go through each of these stages, it takes approximately 18 weeks to get through the first stage, approximately 24 to 48 weeks to get through the second and third stages. So, less than a year overall to bring a project to fruition for sure. But to your second point, we don't have any fail-safe mechanisms. Our goal is to see these projects fail as fast as possible. It's not very expensive to test these ideas, especially outside the context of any requirement that the experiments be done in a way that creates a novel method so that it can get in a cell. To be frank, I think you'd find it simpler than you might think to test through one of these to see if there's going to be a viable target or to see if the compound is going to be something that can warrant a partnership with a university. So rather, we have fail-safe mechanisms in that we have exit points. We have 3 exit points, right? Which is after step 1, step 2, and step 3, which are groups of experiments that have a certain kind of, we bind them based on their, both their cost and also the requirement for a certain amount of full-time work. And so if we exit after phase 1, we'll have spent $5,000, $6,000. And so in that way, that's our fail-safe mechanism. We're happy to test through as many targets as possible and try to make as many drugs as possible because I can't predict how, he can't predict how, and anybody who tells you they can are making it up.
And a follow-up question for that is that you will hear this a lot from academics now that we have cured enough mice. When are we going to cure human beings? And I think that is one of the biggest challenge for us is that we have targets, we have identified the mechanisms. When we want to go a second step, it's generally not happening. Is there a pipeline that you are thinking of that can sort of recapitulate some of those disadvantages of just having an animal model?
So this is interesting. I mean, I personally have, can you hear me? Yeah, so I personally have read the literature into this, and there are some scientific strategies that people are taking to try to predict this. In my view, I kind of see it as misguided because people are never going to accept those predictive things, right? Even if you came up with something in a computer or on a chip, a human on a chip, people are still not going to be comfortable with taking it until it goes in people, right? So to us, our entire model is trying to reduce the cost of trying. Right? The cost of preclinical research, if it came down, would also solve that problem, right? Because fine, people are sick of curing it in mice, but do it 5-6 times and you'll cure it in humans. But of course, you can't say that to somebody who took 5 years out of their life to do it. So increasing the rapidity of the project and also reducing its cost is how we handle that.
So to get at that a little more as well, so there are a lot of cases where there are things that are validated in academia, but then never are acquired by larger companies. And so this is a lot of what led us to start Drug Shop, is this observation.
This is something that I can share with you guys that might be useful. I've had many discussions with individuals at large pharmaceutical companies about what they want to see before an IND. I know that a lot of you probably are in the protein ligand space or essentially in some kind of biomedical space. They really have much fewer requirements than you might imagine, but they're pretty specific. They want to have a gold standard view of binding affinity. You need to know the target is and that it's hitting it. Additionally, 1 or 2 very good preclinical models of efficacy. So after you know the target and that you're binding it, do you have this measures of efficacy that outperform other measures of efficacy to a similar disease, therapeutic area, or target? And third, have you done significant safety testing? And generally, a laboratory will only have 1 or 2 of these 3. But if you have those 3, that's when you can really move forward. So I think you don't necessarily have to have a big, complicated cohort of pig studies to move forward, but you really need to have some focused studies that tell them what they want to know. I know there's a question back there. Oh, sure.
So how do you make your connections with the big pharma companies?
Just call them. So large pharmaceutical companies, so there's a new kind of, this is really only developed over the past few years. Johnson & Johnson has something called J Labs that's right here in Houston, and they're one of the top 10 largest pharmaceutical companies in the world. Just walk over there, talk to them, and introduce yourself, and then say, there you go. You'll have met them. I mean it. And they'll direct you to somebody that you can talk to. If you want to talk to somebody at Eli Lilly, make sure you talk to the external innovation department. At Sanofi, it's called Sanofi Sunrise. You have to go look for the externally facing research components, which exist for at least all of the top 10. They want to talk to you. That's been my experience.
If you had a compound, they'd probably talk to you tomorrow. They're very seeking of this kind of thing.
And they'll say no quick, too, right? Certainly I don't think that no one's going to sit down with you and have a 3-hour meeting until you really have several calls and some solid data, but people won't ignore you if you haven't wasted their time. Oh, sorry, yeah. You guys control the questions, I'm sorry, yeah.
So this sort of gets back into your kind of no-go, no-go, where you're going to cut off the compounds, fail early, fail cheap sort of philosophy thing. So specifically, I work in bladder cancer, and as a disease, it's incredibly heterogeneous. If you're taking, if you cluster tumors based on RNA expression profiling from the bladder, they will cluster with other tumor types a lot of the time before they cluster with bladder tumors. And so when you're trying to understand what what the right model is, what are some of the criteria that you go for when, okay, so we have this compound and it's not working in this setting. Do we throw it away, or are we approaching this the wrong way? How do you think about that as a question?
Great question, and I hope this can be useful too. So the due diligence process for us, it's a little bit different than the due diligence process for our end user, the pharmaceutical companies. But I have a pretty, like I said, there's these 3 aspects of the target, the efficacy, and the safety, right? And so if you have efficacy failure, right, you're saying, how do you know that you really have enough efficacy failure to abandon the project, or maybe you can change something about the molecule, right, or about the approach? How can you pick that? Is that the question?
So I'm thinking more of talking about if you're looking at a disease where you might have, you have one phenotype, but there are 5 different things that could be causing it. Am I working in the right model? So I'm hitting what I want to be hitting, but the reason it's not working is because I've picked the wrong PDX model, or I've picked the wrong mouse model, I'm working with the wrong cell lines. So how do you sort of work around that, or how do you think about that when you're designing your experimental planning?
So maybe, hopefully this helps, but I can tell you that what we do is look for gold standard models, and I know that's not very creative, but so for a specific disease like bladder cancer, right, I know for like non-resectable, the treatment-resistant bladder cancer, there's a certain group of cell lines that are commonly used, a certain xenograft model that's commonly used. And so for us, we really look at, because we are selling preclinically, there has to be a demand already, and if there's a demand already, there's a therapeutic area. And if there's a therapeutic area already, then there are standard disease models. So I would do the literature review to see if what has been used to validate other therapies, because you want to beat them. Mm-hmm. That's really, so if you're going against a new therapeutic area, in addition to it being a new therapy, that's a real challenge. And I think that it's not where we place our focus, but we tend to look for what is the current standard of care been tested in? How do they test those measures of preclinical efficacy? Because if you can just beat those, then you're in the right space.
So, I'm sorry, just one more follow-up to that, and I'll then pass the mic off. But, so do you avoid then spaces where the models are, from what you can tell, underdeveloped, which is, again, a problem that we have in bladder cancer, where we don't have our KRAS mouse, we don't have a PYMT mouse like they have in breast. It's not a space that people have been working in very much for a very long time. Not that there haven't been people working, but not a lot of energy has been thrown at it. And so that's great, because there's a lot to be done, which from a company standpoint can mean that you can make headway and you can get something that might end up filling a niche that's not met. But that also means that there hasn't been all of this back work to set up the stuff that you need to test. So do you avoid those spaces, or are they actually something you might want to go into, given the fact that there's more room to make progress?
We avoid those spaces, right? Yeah. Because we're a small molecule company, but startups shouldn't. For example, a very common licensing approach, your technology transfer office will love this. If you do come up with a new model that can be licensed, right? And then all of a sudden a pharmaceutical company needs to test their drugs through your model. That's also a commercial opportunity. And so in that way, I think that we see that as part of the drug development process. It's just not if the model is not there, we can't make a small molecule. But if the model is not there, you should see the model as a commercial opportunity. It can be a startup, it can be a license, it is a technology. And if you develop it and you protect it, and you have some barrier to somebody else using it, then at least for a little bit, you can recoup the costs that it took you to do all that back work. We have immense respect for that back work, right? And it's just that if it's not there, we can't find a small molecule.
Yeah. So just going back to what you said with regards to the product value and the product inventory for an early stage startup, I think you mentioned that your company has a diversified portfolio of products. And while that would probably be a better move and it's much safer because you never put all your eggs in one basket, can you leverage things like economies of scale? Can you leverage actually putting everything into a product before it's failing? How do you manage to actually just balance the two out?
So can you repeat sort of the crux of your question again? I'm so sorry.
So it's just how, as an early stage startup, I would think that you might...
how do we balance risk?
How do you balance risk? How do you balance having multiple products and at the same time giving each product everything that you can?
Sure. So I think
economically,
a really good thing to say is you won't have all of our attention all the time, but you'll get it when it counts, right? Meaning that these projects happen... Every professor, maybe I'll give this one to Corwin. This is a thing that PIs have to manage all the time, right? Multiple projects at once.
So this is rather similar to having multiple projects in a lab. So I'm sure many of you have the experience having your project be scooped and realizing that you have to come up with a new project, and it's hedging your bets by having more than one at once. And so you often have a case where a project is being set off to be sequenced, or there's some kind of delay step which lets you work on the others. In our case, we can, much like a lab, have multiple people working on multiple projects. And so, it's the same way you manage it in an academic lab is the crux of the answer, I suppose.
And one thing we're really trying to do, we're trying to get into a step, once we have more partners, this will become more critical, but get into a step in which our laboratory this week is doing work on Project A while our collaborators are working. And then next week, we'll be working on Project B, and we'll get back to Project A. And then basically, our collaborators are kind of functioning as our company in a way. And they all stand to benefit from it. But in a way, we try to expand the diversity of what we do, not just through ourselves, but also because we know that when we're not working, our collaborators are.
Maybe to answer that again as well. So in certain cases, we have one assay, and we can more readily pool projects if we have to do the same assay, which might drive your question of that as well. So if we have to do custom chemical synthesis and we're ordering a batch, we might add multiple projects at once. We're doing a binding screen. We can do more than one target as well.
That's a good point. It might appear like we have no focus, like we just have all these different diseases, but the fact is we're actually very tightly focused. We just have a few assays that we do. We have a few computational models that we use. It's really just, to us, our partners focus on the aspects of the disease often that have to do with their specialist expertise. And so bringing it together in the form of a molecule is what we view as our specific focus.
Hi, do you have competitors in this space? And can you talk about the timing of starting a startup for you personally?
Sure. So competitors in this space, every pharmaceutical company is a competitor with another pharmaceutical company. Every pharmaceutical company is a pooled, distributed portfolio model. So I guess I literally mean that. Every pharmaceutical company, every tech transfer office, there's the company I used to work for, Fan and Innovation Studio, all these places can be seen as competitors. But the fact is that in this industry, this is inelastic. My success doesn't hurt anyone else's, right? It's not zero-sum in the least. If two people invent two different cancer cures, it's not like, oh, we're not going to go on this one. It's not exciting enough for us. It didn't save enough people's lives, or it doesn't have enough preclinical promise. There's no single set amount that they're going to spend per year. It actually varies quite much how much each individual pharmaceutical company spends on preclinical acquisitions per year, because it kind of comes as the opportunities come. So we view our competitors as also our like partners. In the end, if we all sign agreements, then we'll all stand to benefit from it. But this isn't something in which it's not like a snowplow. It's not like that famous Simpsons episode with the snowplow competition. If you plow somebody's driveway, there's no more business. It's not like that. Our competitors are other pharmaceutical companies and other licensors of preclinical technologies, which would be tech transfer offices and the like. The timing? So yeah, the timing was interesting. So we, I guess, started working on doing the background work to develop the models and the research that we needed last year at the end of 2016. But we really didn't get going, so we went live in January of this year. That's when Dr. Miller joined me and we really began our operations. We began partnering and doing our wet laboratory research as well and applying those models. And so it's been about 4.5, 5 months.
Do you guys know each other beforehand, or how do you identify partners? For example, the IP part.
Sure, so we know each other, but partners, we identify partners through their, of course we, like everybody, are working with individuals that we met through our careers. They're the best, most safest type of collaborators to start with, but we're open and we're now working with people outside that realm as well. We generally seek out partners ourselves. We'll contact you, but we're open to other people attempting to partner with us as well. We really only want to work in areas where we need their specialist expertise.
Just one more, sorry. How important is a PhD degree in terms of starting up a company in the biotech business?
Sure. So I know this topic real well. And I'll say that it's critical to have somebody in your pharmaceutical company that has a PhD, because how else are we going to apply for grants, right? It's a legal requirement. But I'll say that personally, nobody that is real, it doesn't really bother anybody who's informed to not have a PhD, right? Because essentially, in my experience, most people with PhDs are the kind of people who are smart enough to evaluate people on their know-how. They make fun of each other. So I would say that it hasn't really hurt that much, though I would say that with some people, especially investors, it can be seen, especially when I was alone, it was difficult because they would prefer, or they would kind of go towards the general case of a early stage pharma company, which is like a professor, and of course they have a PhD.
Okay. So I know you just started this early stage, but where do you see the company going in, like 5 years? Like, what's your long-term plan?
Our long-term plan was that it could be an engine for the Texas Medical Center, because we would be able to, our 5-year plan is to integrate more, to get more interns in the space. We don't even really call them interns. We would really just want to get consultants from the graduate student and the postdoc pool to work with, find laboratories to work with in the medical center, kind of expand. What we want is to be doing about 24 projects a year. And we want people to be willing to start these projects, low threshold, not consider them to be a massive deal. So we really want to start moving the needle in this community, in the Texas Medical Center, to start to understand that we can all benefit from the research and it can all be done preclinically. We just have to, don't put all your eggs into one basket, and have a mechanism for that early stage commercial development that professors can readily access, and that people and projects that can be done without the fear of not getting funded again. So we want to be able to offer small projects and cooperative growth in the Texas Medical Center.
So you'd like to have your own space, like finding innovations?
Yeah, we would love to have our own space, right? And we're working with we'll probably have our own space in the next couple of months.
So one thing I'll add is that finding new collaborations has not really been the challenge for us so far. So in the years to come, what we would foresee is forging more collaborations, starting more projects, and hiring more people.
Yeah, so looking at your pipeline, what happens after step 3? Where do those...
Sure, so let's say we've secured the composition of matter IP. So that's really what we consider to be ours, the composition of matter IP, right? That's really where we want to get. And of course, there's a basket of IP and data that comes along with that. So if we have that IP and we have some small animal toxicity studies, we will approach the various external innovation departments at pharmaceutical companies with our data. And so if that data is really crisp, and like I said, if it has the triad of efficacy, target info, and safety, you're going to get looked at. And if it's a therapeutic area that's important to them, then they'll call you back. And so this would be something where I would make phone calls to individuals who I knew had interest in the therapeutic area and simply email it to everybody. And you should be willing to give this data out broadly at the time it's going to be commercially viable. You should never send just a whole bunch of data to someone at a pharmaceutical company because that's it. If you send them a whole bunch of data saying, I have this drug, I have this research, I have this data, you just gave it to them for free and there's nothing wrong with them taking it. So you have to be very careful about this. And so that's my only real advice. But what we would do is I would make a special effort to contact those who I knew had an interest in the therapeutic area, and I would email everybody I knew.
So that would be your company's products?
Yes, the basket of IP and data. That's the product.
So how do you vet potential collaborators? Because if you end up working with somebody or another lab somewhere, you're effectively, in sort of a weird sense with your business model, like incorporating them temporarily into your company, into your pipeline. So how do you look at somebody and know whether what they have to offer is something that will actually be useful, and more importantly, whether they're actually capable of delivering what they claim to be able to deliver?
Sure. So, we work with specialist experts generally. We're not looking to work with labs that are the super labs, right? So we can always assess if they're capable of doing it by looking at their research, right? Similarly, same thing for us. They might ask, we'll sign an NDA, look at each other's research. There's a way to do it. The things that are important to me, like you said, are very similar to that. There's just, can they do the work that they claim to be able to do? And are they willing to split the revenues in a way that's fair? Because not every project is even close to the same amount of work. We may only need a collaborator for one small thing. So how do we vet? What's important to us is more, I can trust almost always that most research groups, most underfunded research groups who are still beating the wall as hard as they can for their specific specialist expertise, they're great partners. Not only do they need you and we need each other, but they obviously really care about what they're doing. And so I try to not, I don't like look into people's life backgrounds, but we kind of look into their research and then we assume that let's move forward. I think there's certainly not much of a downside to partnering with for us. If we don't like a partnership, it'll just end.
So one thing I'll add to that is if they don't invent any IP, then they don't invent any. And so worst case scenario, if they don't do anything, nothing's really lost by forming a new collaboration.
Okay. So sort of a follow-up to that is, I know that with a lot of the larger sort of firms, and I'm talking about somebody that can have their own entire R&D department, like Pfizer, AstraZeneca, there's been, with some of them, an effort to pull some of their basic research in-house. I know that for a really long time, they would rely on the academic community to sort of generate the low, I guess you call it low yield, not immediately leading up to producing pharma, like IP level stuff, and they would take it and build off of it. And after a while, it got to the point where they were tired of getting burned, where there were labs that were putting things out, not necessarily labs specifically, but stuff that just didn't end up being reproducible, and they could throw, millions of dollars and a ton of time at something, and the underlying premise just wasn't there.
This is a great question.
Yeah.
So large pharmaceutical companies are convinced that everything in academia is fake. They seriously, I think it was a Merck in combination. Don't quote me on that exact one, but there's a major study of two major pharmaceutical companies that found it was a significant percentage, maybe 60-70% of the findings. I think I'm lowballing this number actually, was not reproducible. So, however, I have to disagree with one thing you said. You had said that they're trying to bring that basic science in-house. Yeah, there's both problems. They're not trying to bring it in-house. It's too expensive. The Sanofi-Aventis CEO in 2015 famously stated in the investor report that my job is to never open another R&D center, and they're just closing them all by attrition. All the numbers show that at least at the top 10, the amount of basic R&D being done is collapsing in the past 5 years. So they're stuck in a rock and a hard place. This is why we opened Drug Shop, because we instead know that there are plenty of labs that aren't just putting out, work that is either overblown or maybe, making conclusions too quickly. We simply view our role is to look through and find that. So part of it is to seek out the really good research by redoing it. We require everything to be blinded. If you aren't willing to blind every experiment, you can't work with us. So I guess there's one way that we work there. We blind everything, internally, externally. Why not? You're just lying to yourself if you don't. And so to us, we view part of our role is to act as that filter for them in addition to doing the research. So that's how we would handle that.
And so, to go with what Errol was saying, so there's been report after report over the last decade of how the efficiency in R&D in large pharma companies is going down. So basically the cost, the dollar cost per new drug just continues to climb. And essentially the way they've dealt with that is just by outsourcing more and more of the R&D. So they want, essentially it looks like they want their task to begin at the phase 1 clinical trial and to acquire everything that would come before that. They don't, it's not 100% yet, but that is the direction they've been steadily moving.
And it might be interesting to note that when we say outsource R&D, it doesn't mean that they're paying labs to do work. They're buying and in-licensing technologies. They're paying for that work. They're providing this end prize, right? And so again, this gets to the crux of if pharma is having a problem doing R&D and academia is having a problem funding R&D, what's going to meet in the middle? And we try to help meet that in the middle a little bit.
Thanks so much for your presentation. I ask one question two different ways. So what advice do you have for the current grad students, postdocs, what resources they need to use, what skills they need to develop or improve if their next career chapter is meant to be starting their own startup? The other way asking this question would be, you're expanding in the near future, you will be a potential employer offering internships and jobs. What are you hoping to see on application packages, either as a partner or employee or intern? Thank you.
Sure, I'll give you I'll leave one of this to you, sorry. Okay, so to answer your first question, to bring up the first question, what kind of skills do you really need to start a startup? I think I'll defer to Dr. Miller on the challenges that you might face going from, say, a postdoc to running your own laboratory immediately, which is what's going to happen if you run a startup. But from the other non-scientific aspect, if you want to start your own startup, or if you want to commercialize your own technology, your first and primary concern should be, how am I going to derive a profit from it? And the fact is that might sound crass, but it isn't. You have to start there, meaning it's just step one, right? You have to see it as step one, meaning can you work with your tech transfer office to make a deal? Can you sign agreements with another partner of yours or a professor? But if you can't see a path in which your technology will be able to make money at the end of it, don't start, right? That's something that's really, really critical. That's my most critical advice. And it sounds crass, but it's the truth. You won't be able to move forward without it. So second, if you want to develop skills, I would go to JLabs. I would seek out their classes and go to almost every one. They're fantastic. Station Houston is another place that has a lot of resources on how to get mentors for helping you with some of the aspects you may not have been taught in graduate school, like financial accounting. How do I start a legal structure for my business? Can I start a business while I'm in grad school or a postdoc? Yes, but there are ways to do that. So I would seek out TMCX is also another place that has great resources. These classes occur in TMCX, JLabs, and Station all the time, on honestly a daily basis, and they're often freely available and always come with discounts for people who are postdocs or graduate students. So that's the first thing I would do. And you should also develop one skill that's really critical, and that is that you don't want to take advice from anybody, right? Not everyone starts a startup. Very few people actually begin their business from scratch, even if they've been part of one. The knowledge space is very limited and narrow, and you'd be surprised at how much you can actually do that you may just assume that you can't right now. So don't take no for an answer, and listen carefully to a lot of different people. But don't just go to one person and say, can I do this? And they'll say, no, you're not allowed to do that, and then come to the conclusion that you can't. This is the number one way I see graduate students and postdocs frustrated. They think they can't commercialize their technology, but they can. They think they can't negotiate some way for it to be viable, but they can. You have to be able to keep pushing what it is that you want to do over and over. Now, there's a big stark difference between being a postdoc and a graduate student and running the laboratory, and I'll let Dr. Miller talk about that, and then I want to talk to you guys about job opportunities.
So in a lot of ways, it's similar to going from being a postdoc to being a professor. So you do have to run kind of a small batch of people and manage the ideas and drive the projects. But there are some key differences between being a professor and starting a small company. Name a few. A large institution has a tech transfer office. We have to do all the tech transfer, everything ourselves, essentially, at the start. A large institution manages the accounting and the payroll, and of course, we have to do that ourselves as well. So overall, there are a lot of similarities, I'd say. It's more similar than different to becoming an untenured professor, just with a few more things you have to do yourself.
And for example, we were able to get Dr. Miller immediately as an ERA Commons PI, set up our institution, get all of the same kinds of registrations with the NSF, the NIH, and NIST as a tenured professor would. But the skill ended up really being going through and asking and making sure that we're doing the research to be able to find out exactly what do we need to have in place, because it's like going to a place. It's like, imagine if you got a tenure appointment and then there was nobody else in the department but you, and also you had $50,000 and you had to figure out everything. This is really a startup, but to his credit, he was able to do it, and we have a multi-institutional R1 going out tomorrow. And you would be surprised, something very critical. For every postdoc in this room and every graduate student in this room, remember, if you are the PI on a grant within 10 years of graduating from your PhD, you're considered to be an early stage investigator. If you're an early stage investigator, you get a bump anywhere from 1 to 10 percentile points on the grant score. It's incredible, and it should incentivize and allow you. The point of it was to incentivize people to go off on their own earlier. And so a lot of individuals may feel that they need to continue to get that experience by doing a year after year of postdoc before they're capable of doing their own, becoming a PI. But the NIH feels differently. The NIH wants you to become independent early, and it gives you mechanisms to do it. There's actually grants that only you can apply for 10 years after you get your PhD. So if it's still been 10 years since you got your PhD, please look into this. The Early Innovator Award, ESI status. The NIH has mechanisms to throw money your way, and you should use them, because that's the only way the investigator's average age will go down. And if there's any lesson from this, is that you can still be an investigator and be part of a startup, for sure.
So you can also ask what we'd be looking for in employees. So one thing I'll add is that a complaint I've heard from others in the past is when they join a large company, a lot of the creativity is stripped from their task. They're stuck doing the same sets of assays essentially a million times. At a small company, I think this is very different. We would certainly want someone who's independent, who can drive projects and largely can tell us, as much as we tell them, ideally.
And in terms of opportunities for that, we are hiring. So, for example, we have gotten our first. We had one intern. It was an MD Anderson postdoc that will remain nameless. She didn't want her PI to know, and I'll respect that. And we are still hiring more. And what we are looking for are individuals to be involved in any one of 3 aspects of drug development. That could be on the licensing and partnering side, computational modeling and medicinal chemistry, SAR, that early coming up with the compounds to test, and then also testing the compounds, assay development, testing on preclinical models, XTT assays. So we need people for all 3 of these aspects. And we know that there are opportunities out there in the community to get similar internships. And the one unique aspect that we offer, of course, we do pay anywhere from $15 to $20, depending on the skill, experience, whatever level you are. We also offer you incentive compensation, meaning that you will receive a royalty or some sort of bonus from if we do end up using the intellectual property that you develop. We won't see anybody invent something and not get paid at this company. That's a key that's a key value for us. And if you do decide to work full-time, every full-time employee owns at least some percentage stake of our company, which is another, I guess, uniform benefit of a startup in general. And I'd love to talk to you guys if you're interested in starting. We are hiring right now, and we certainly could use the help. Any other, yeah.
This may be very specific questions, but I would appreciate if you can just give me an overview of it. So I know that some of these compounds will be targeted to proteins, but do you have any compounds which absolutely target just RNA, like non-coding RNAs and microRNAs?
You know, we don't, I guess we won't go into exactly the extent of our pipeline, but right now we don't have any particularly active RNA projects because we specifically decided to start up in the small molecule space because it's the least expensive, most successful way to do it. It's just that biologics, by their nature, are less stable. They require a higher, believe me, pharmaceutical companies want to know way more about your RNA than they would about a small molecule. Way more about an antibody than a small molecule. Of course, the dollar sizes for a deal is much larger, but I mean, it's not even close. You're going to need millions of dollars of research for each project in a biologics project.
Sorry, I didn't mean about using biologics to target. I mean your small molecules can target RNA.
Oh, I'm so sorry. Yes, like many antibiotics do, like ribosomal sites and stuff. No, none of our current ones do. But I'd be definitely open to that because we do and can. We have the force fields to model RNA, and we do model RNA, and RNA is predictable. So I think we would love to get into it if that's something you want to do.
Any other questions?
Yeah, so I had a question. Would you guys or companies like small startup companies value like postdocs who had tech transfer, like pass the patent bar and stuff like that? You would look favorably upon that, probably?
Of course, yeah. So we have three aspects, and that's one of them. Absolutely. Of course, we'd probably come up with a different compensation model, but yeah, absolutely. I mean, I feel like it's certainly something that we would benefit from, and to be honest, if there's anything that we need that takes the most amount of my time, it's that. And so being able to bring on individuals with some background experience in it to help, and that could immediately help, would be incredibly useful for the company. I'm sure there's a way we can work it out. So yeah, I know it was a little bit different than most of the other presentations that you probably get. I hope it was useful to you guys. But you know, starting a startup is a live experience. It really is, and so you know, it's one of those things that I hope that there's some people in this room who want to have that experience and come on the journey with us as well. Thank you so much.